Hi everyone, today's screencast is going to cover plant stem cells. Please make sure you have a pen and some paper before we start. Once you've got a pen and some paper, write down today's LO, which is to understand plant stem cells and their uses. Once you've written down today's LO, have a go at these retrieval practice questions from cell biology about stem cells. Make sure you pause the video while you have a go at these questions. Once you think you've done those questions, you need to make sure you've got a different coloured pen, and we're going to go through the answers. So what is a stem cell? They are undifferentiated cells that can differentiate into one or more specialised cell types. Adult stem cells are found in the bone marrow. The other type of human stem cells are embryonic stem cells or stem cells from an embryo. An advantage of using adult stem cells is that there are fewer ethical issues involved. They're obtained from adults who can consent to their use, whereas embryonic stem cells are obtained from an embryo which obviously can't give consent. A disadvantage of using adult stem cells is that it can take quite a long time to find a suitable stem cell donor. They can only differentiate into some specialised cell types as well, so they treat fewer diseases than embryonic stem cells. An advantage of using embryonic stem cells is that they can differentiate into any type of specialised cell, so they can treat lots of different diseases. They are also easier to obtain because they're found in spare embryos from fertility clinics. A disadvantage of embryo stem cells is that there's ethical issues surrounding their use. Every embryo is a potential life. There are also potential risks involved with the treatments, such as transfer of viral infections. Please make sure you're marking your questions as we go through them. Make sure you're also correcting any answers that you haven't got right. You will need this information later in the screencast. So we know that stem cells are undifferentiated cells that can differentiate into one or more specialised cell types. Stem cells are found in both plants and animals, so there is another screencast on stem cells in animals, in particular in humans. This screencast is going to focus on stem cells found in plants. So we can get animal stem cells from bone marrow, adult stem cells, and embryos, which are embryonic stem cells. In a plant, we get the plant stem cells from a region called the meristem. So the meristem is the name for stem cells in plants. Plant stem cells can differentiate into any type of cell throughout their life. So whereas in humans, our adult stem cells can only differentiate into a few types of cells, plants keep that ability throughout their life. So their stem cells can differentiate into any type of cell throughout the whole lifetime of the plant. We can take cuttings from plants and make clones of those plants. So the apical meristems are found in the shoots and also in the roots. In the shoot tips, they encourage the shoots to grow towards the light. In the roots, they encourage the roots to grow down towards water. There are also lateral meristems shown here. These cause secondary growth, and this is the widening of the plant. So if you imagine the layers of bark on trees, they cause a plant to widen. So if we look at plant cloning in more detail, it's carried out by a technique called micropropagation. During micropropagation, small pieces of a plant or explants are grown in vitro. This basically means outside of the plant. So they're placed in a petri dish using sterile agar jelly containing plant hormones and nutrients, and this allows the plants to develop, so the explants develop into plantlets. And this produces a large number of plants which are clones of the parent plant. So these plants will grow and they are identical to the parent plant that we took the tissue samples from. This is an important way that we can preserve rare plant species. So we can actually stop plant species from going extinct. If we have a few individual plants left, we can clone those individual plants to create a large number of clones. It's also used commercially to produce large numbers of genetically identical plants with desired characteristics. This can be done any time of the year. So farmers, if they have a plant that has a really high yield, they can actually clone that plant and have lots of plants with a high yield, which increases their income. Unlike in animals, plant stem cells can be obtained really easily, and there's no ethical objections either because we are taking them from the meristems of the plants. They can then be used to produce clones of plants quickly and economically. Plant clones can be produced in weeks at relatively low cost for the potential value. So like we said before, it won't cost the farmer a lot of money to clone their plant and have lots of those plants with potentially higher yield. Rare species at risk of extinction as a result of human activity can be cloned to protect them and maintain the biodiversity in an area. The plants that are produced, though, are clones, so although the number of plants increases, the genetic diversity between those plants is going to be low because each clone is genetically identical to the parent plant. So they're all going to have the same genes. When we do clone the rare plants, though, it gives scientists a little bit more time to study them and they can see if they are going to produce any compounds of interest. So they might be things that we could even use in medicine. As well as things like high yield, crop plants can have special features like disease or pest resistance. These can also be cloned, so we can produce large numbers of these plants, and it would reduce the need to use chemicals like pesticides that would kill the pests that live on plants, and this will have environmental benefits if we aren't relying on those chemicals as much. So what I'm going to do is pop a few past exam questions on the screen. I want you to pause the video on each question and have a go at it, and then I will go through the answers at the end. So here's the first question. Here's the second question. And here's the third question. And when you're ready, we'll go through the answers. So make sure you've got a different coloured pen. So question 1 says, as a plant grows, new root hair cells are formed from unspecialised cells. How does an unspecialised cell become a new root hair cell? The answer we need is differentiation. So cell differentiation allows unspecialised cells to become specialised cells like root hair cells. The next question says, scientists can clone plants using tissue culture. Figure 2 shows the process of tissue culture. So we saw a diagram similar to this before. Why might scientists want to clone plants? We can either have to create new species of plants, to introduce variation into plants, to protect endangered plants from extinction, or to reduce disease resistance in plants. We know that we can clone plants to protect endangered plants or rare plants from extinction. The last question says, what is the advantage of cloning plants using tissue culture? So we could have no special equipment is needed, plants can be produced quickly, all flowers are different colours, and the offspring are all genetically different. We know that the plants are produced quickly when we clone plants using tissue culture. Make sure that you have corrected your answers. If you've got them right, make sure you've ticked them so that you can use the answers to these questions and your notes from today's screencast as revision.